Hello and welcome to day 2 of the Greenlight Guru True Quality Virtual Summit, the 3-day event designed to provide actionable takeaways that you can implement in your own company to innovate faster, stay ahead of regulatory changes, and use quality as a strategic asset to grow your device business. This session is on how recent FDA feedback regarding extractable and leachable testing and how to make sure FDA will accept your testing protocol. My name is Jessica Lyons. I'm a senior medical device guru here at Greenlight Guru, and I will be your moderator for today's event. We've got a really special session scheduled for you today. I know our speakers, Matthew Jorgensen and Thor Rollins, are really looking forward to covering one of the newest biocompatibility evaluation tools, extractable and leachable testing, and the best way to make sure you are performing the correct version of the test by analyzing the most recent FDA feedback on studies. Before we dive too deep into today's presentation and introduce our speakers and their company, Nelson Labs, I'm going to touch on a few items really quickly. First, this session is going to run for about 45 minutes in total and will include a question and answer session at the end where Matt and Thor have been kind enough to answer all of your guys' questions. So I encourage you to submit those throughout the presentation as they come up in the box on the right-hand side. We'll get to as many of them as we can. This entire session will be recorded, and Matt and Thor will follow up with you guys afterwards regarding any questions that don't get answered. Once this session wraps up, there is a 30-minute break before the next live session begins. If you're interested in learning about IVDR readiness, you will want to be sure you're registered for that next session. Use your unique link to tune in. Don't worry, if you aren't already signed up, you can register for the next session and over 20 others at virtual-summit.greenlight.guru. Okay, I'd like to go ahead and share a few words about Greenlight Guru and why we put on this free virtual summit. If you've been on one of our training sessions before, then you know we put these on because improving the quality of life is our mission here at Greenlight, likely a similar mission as many of you at today's summit. Anything we can do as an organization that helps device makers bring safer, life-changing devices to market quicker and with less risk aligns with that mission. We're constantly looking for ways to fulfill that mission, whether it's through hosting free events and training sessions, through partnering with world-class medical device consultants, or through our award-winning medical device quality management software. If you'd like to learn more about why medical device companies from across the globe are moving away from paper-based general-purpose quality management systems and adopting our purpose-built medical device quality management software, I encourage you to head on over to www.greenlight.guru after today's presentation to schedule your free personalized one-on-one demo. Now, on to the bulk of today's presentation. Let me give you a proper introduction to today's speakers. Dr. Matthew Jorgensen is an expert in chemistry and material science who works at Nelson Labs, the leading provider of global lab testing and expert consulting services. He has over a decade of experience designing, synthesizing, and analyzing complex materials. His analytical chemistry background includes research in organic chemistry, synthesizing and analyzing a naturally occurring anti-cancer drug, and computational treatment of photothermal spectroscopy. Most of his materials research has focused on the intersection between chemistry, materials, and physics, fabricating structures with special micro and nano patterning to introduce novel functionality. His research has resulted in over 30 peer-reviewed publications. He is joined by Thor Rollins, a certified microbiologist, and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility-related topics through Nelson Labs' external seminars, webinars, and trade shows. He presented on biocompatibility at the American College of Toxicology annual meeting in 2013 and has many published articles on biocompatibility topics. He is a participating member on all Association for Advancement of Medical Instrumentation, TC 194 and 10993 ISO committees, and plays an active role along with the FDA and regulatory committees, developing standards, discussing biocompatibility methods, and voting on changes to those standards. As one of the select group of experts in the industry, Thor's participation on the committee offers him insight into industry changes and helps prepare clients for changes in testing. So, without further ado, let me hand it over to your presenters, Matthew Jorgensen and Thor Rollins.
Thank you very much, and thank you for joining us today. We are very excited, like always, when we get to talk about chemistry and biocompatibility. It's one of the things that we actually love almost more than anything. And so we're excited to bring these 45 minutes to you today. I just want to start as kind of understanding why we're in the position to be able to do this. As consultants here at Nelson Laboratories, we work with medical device manufacturers all over the world with very unique products, looking at ways that we can develop a risk assessment for biocompatibility or safety of that medical device. And then we work with regulatory bodies, including the FDA, notified bodies, Korea, Japan, China, basically all you can think of. We work hand in hand with them, with these medical device manufacturers, to try to come up with solutions to make sure these medical devices are safe. Part of that is getting feedback from testing or proposed plans that we have, and we interpret and take those FDA responses or notified bodies responses, and we are continually improving our offering to make sure that we are meeting those requirements. So with that, we constantly talk with the FDA on a daily basis sometimes, where we are discussing the scientific approaches for extractable leachables and toxicology to make sure that we are meeting just their needs and also our clients' needs. And so with that, we decided we would take some of this recent learning, since this is a very new area for biocompatibility, and it's very dynamic. We're seeing huge changes through the last few months on what regulatory bodies are wanting in the realm of chemistry and toxicology. So we've kind of collaborated and correlated those responses, and we're going to present to you today kind of the high level what we're seeing from the FDA. Now, there's no way that we're going to be able to give you everything in 45 minutes. In fact, we do these for clients, and it takes sometimes 3 hours up to a day to go through all the nuances of what the FDA is expecting. But we've taken the hottest topics and the biggest impacts, and we boiled it down into 45 minutes. And we're going to try to share those with you today. So with that, I'd like to turn the time over to Dr. Jorgensen here, and he'll kind of step through what we're seeing from the FDA recently.
Yeah, sounds great. Like Thor said, I think that we're both very, very excited. It's almost a little bit embarrassing how excited we get to talk about this stuff, because it's so dynamic and so cool, and we get to help people with medical devices keep their devices safe and get through the FDA as painlessly as possible. So it really is a lot of fun. So I think we like to start with this slide. It shows a small part of our consulting group as it is right now. That's me there in the middle, and you can see Thor, of course, there to my right. And on my left-hand side is Dr. Sarah Campbell. She's our head toxicologist. Trevor Fish there is another toxicologist, and Audrey Turley behind me is another biocompatibility expert. And one of the reasons why I like to show this is to highlight or to emphasize the fact that if you're going to do an approach to get your medical device through the FDA that includes chemistry and toxicology, you really need to have this trifecta of experts, like three legs of a stool, come together to provide support of that project to make sure that it's done right. So it requires the feedback of biocompatibility experts, chemists, and toxicologists. They all should have their hand in the plan to make sure that all the needs are met. And where I've seen things go poorly is when one of these groups in isolation makes a bunch of decisions and a testing plan is executed, and then at the very, very tail end, a great toxicologist like Dr. Campbell says, well, it's good work, but we're missing this little bit of chemistry, so I'm sorry, I can't make any claims. And that can have like really tough consequences at the end. Or if you get down to the very end and you've submitted to the FDA, and the FDA's toxicologists say, well, this is great, but the sensitivity was only 100 micrograms per device, and we needed you down at 1.5, then that can be really painful at the very end. So to prevent those types of things, it's critical to have people like Thor and I and others involved in the planning stage to make sure that there aren't any surprises. So just as Thor said, this is really high level. We're going to go through this pretty quickly. We can see your questions pop up there on the right. So if there's an urgent question that comes up, we'll pause for a second to address those. I see that Bob says it's great to see another Mountain Dew lover, and it is true. I am a Mountain Dew enthusiast. We can talk a little bit more about that another time. So let me just give a brief overview of what we want to talk about. We'll briefly discuss what chemistry for toxicology is all about. We call that ChemTox. We also will just give some of the most significant feedback that we have received from the FDA recently. As Thor said, this has been really, really dynamic. There's been a lot going on in just the last couple of months. And then we'll talk about how 10993-18, that's the new standard governing chemistry, and FDA expectations overlap, and also kind of focusing a little bit more on how the FDA and that standard are a little bit more different. So just to give you a brief overview of what chemistry for toxicology is all about, if you go to submit a medical device to a regulatory body, either in the US or globally, you need to demonstrate that that medical device won't cause undue harm to the patient that's being exposed to that. And experts like Thor have worked for decades to determine how those different types of risks scale with the type of device contact that you have. And there's a list of biological risks that should be considered depending on the nature of the device. And those risks are outlined in the ISO standard, ISO 10993-1, which Thor sits on. And so using 10993-1, there's an annex A1, and there you can find all of these biological endpoints that you see on your screen that might need to be addressed. Now, some of those could be considered systemic toxicities. I highlighted those in green. Systemic means that it's related to chemicals that might come off of the surface of the device or leach from the device and cause an adverse effect related to being distributed in the body. And of those which are systemic, the FDA and other regulatory bodies have specifically said that some of those could be addressed using chemistry and a toxicological risk assessment. And it's a little bit more than just saying that it could be addressed that way. Oftentimes, it's a lot better to address it that way because chemistry is so much more sensitive and actionable than the animal tests that would replace. So really, that's what we're talking about. This has become mainstream in the last 5 years, the art of doing chemistry and toxicological risk assessment to address those endpoints that you see on your screen. But what has happened more recently is that while this had very, very wide and like wholehearted acceptance, let's say 2 years ago, there's been a renewed period of intense skepticism on the part of the FDA and other bodies on the details of how that chemistry testing is conducted. And so there's been a lot of scrutiny and change as a result of that. So this is my one slide introduction to toxicology. It seeks to answer a fundamental question: how much of a good thing is too much? And as I mentioned on the slide before, I'm a lover of Mountain Dew. And so what you can see here, so Mountain Dew is clearly a good thing. And this might be a dose response curve for me, so it's very personal. And you can see we have kind of like a U-shaped curve there where if I had no Mountain Dew, then that would cause an adverse reaction. Of course, I would be very, very irritable and have headaches. And then, there's this optimal like zone where there's just the right amount of caffeine in my system. But then as you add more of this good thing, you can see that more and more negative effects start to pile on. So So after a certain point, too much caffeine will start to cause arrhythmias, tremors, you get hallucinations after a stage, and then there's a certain amount where there would be a 50% chance of death if you were to supply that dose to some given population. Then ultimately there's a maximum amount. You can see it's a little bit covered up by our image there, where it's basically guaranteed to, if the negative effect is death, it's guaranteed to kill the entire population. And then of course, going beyond that doesn't increase the effect at all because you've already maxed out. And then there's a whole art to conducting these studies properly in animal systems with the right number of doses and picking those doses at the right level. And usually this results in a more classic curve like this S-shaped response curve, where you get key pieces of data. This is related to the point of departure. This is the dose at which something starts to happen. And you can see there's one dose, the highest dose where nothing bad happens. That's the no observed adverse effect level. This is the highest dose where nothing bad ever happened. And then beyond that, you have other marks along the way. That the lowest observed adverse effect level is the amount that was given to an animal population and something did happen. So it's kind of like a check that, you know, we can even cause any effect on the system. Then of course, on that scale, you have government mandated thresholds. These are based off of the point of departure, and they include additional safety factors to make sure that that these mandated levels are adequately protected for even sensitive populations, young people, and so on and so forth. Okay, so that's what we are really trying to do. We want to generate data using chemistry that we can compare to this type of information in the literature. So that's the scope of the problem. Now when we actually start to conduct that, then we get into a lot of details, and I like to use this real example. So this is an actual medical device. This is a 3D printed spot can, and its intended application is to be implanted into the body next to the heart, so that you can keep the memory of Leonard Nimoy with you always, right? And so, you know, I've been considering this for myself, obviously. And, you know, if I think about receiving this implant and I start to, you know, worry about the types of things that I might be concerned about, you know, yeah, I'm concerned about things that I know might be on the device, right? I know that in 3D printed polymers, especially ones that are photo cured like this, there's often photo activators that can be a little bit concerning and so on. So I'm worried about things that I know might be in there, but I'm also worried about the stuff that I don't know might be in there. And really the concern on patients' part, and really from the perspective of the FDA or our regulators, is they're very skeptical about what we don't know about these types of products. And so the chemistry study needs to be designed to capture anything and everything, both the things that we think might be on there and the things that we might not expect to be on there that would be a surprise. So that really frames the study. It needs to be wide open, a screening study for sure. Any targeted studies, it just isn't adequate for this problem. And to conduct a screening study this way, looking for anything and everything, we use a suite of analytical techniques that you can see highlighted here. So if you take a look at the organic side of things that might come off of a medical device or from the inside of a medical device, we can think of these as small molecules that we would call volatile organics, and then more like medium-sized molecules. These are semi-volatile organics. And then big ones. And for each of these types of molecules, we have an analytical screening technique that's targeted for that. So for small and medium-sized molecules, we use gas chromatography with mass spec techniques. And then for bigger molecules, ones that are so big that they would start to burn or degrade before they can be vaporized into a gas, then we use liquid chromatography with high-resolution, accurate mass spectroscopy for those. On the metals and metal oxide side, then we would do ICP-MS to identify those. And again, we're going to execute all of these methods not only scanning for everything, but we also need to ensure that we have a sensitivity that can prove that there aren't any chemicals above one of these important thresholds that apply to safety. We designed this to extract as many things as possible. We want to make sure that we cover the types of conditions that a device would encounter in the body. So we use a range of extraction vehicles. And this is one of the things that the FDA has been hyper focused on lately. They really, really want to see at least 3 solvents for prolonged and permanent contact devices. They want to see hexane, IPA, and water. If you take a look at the range of polarities of stuff that devices contact in the body, I've given a few examples there. IPA and hexane already are really a lot less polar than anything that a device would encounter in the body. Still, we want to include this broad spectrum to bookend those conditions. So, for example, with respect to these this feedback, the FDA said in your repeat tested, you conducted chemical characterization using exhaustive extraction conditions, and we used purified water, hexane, and then a mixed polarity solvent, 40% ethanol and water. And we analyzed it using the suite of instruments that you see there. And then the FDA said the polarity of 40% ethanol and water is very close to the polarity of water. And therefore, the FDA does not consider 40% ethanol to be a mid-polar solvent. Please repeat exhaustive extraction using isopropanol. So this is a case where, you know, a really rational scientific approach was applied to the testing and the extraction conditions, but the FDA was really digging in their heels on wanting to see what they view as 3 standard solvents: water, IPA, and hexane. And this feedback, or this current position on the FDA's part, it stands in contrast to the new 10993-18, which specifically recommends 40% ethanol and water as an appropriate surrogate for blood and blood-related substances. So it's one point where the FDA is deviating from the standard, and I think that we could say here, and I guess categorically, that the FDA has not officially recognized ISO 10993-18. I've heard, and maybe Thor, maybe you can comment on this a little bit, that the FDA is moving towards a standardized approach to testing.
Yeah, so working or talking with some FDA individuals, they are looking to understand extractable leachables better, just like everybody is trying to understand how E&L for medical devices may work. And that's why we actually have the new 10993-18. A lot of work went into that to try to understand that for medical devices. So the FDA is doing their own work to try to maybe do a standardized approach, like a guidance document, or if not a guidance document, something more standardized that they can put out there so that we don't see variances in responses from different reviewers.
Yeah, and I think that we all of us that are involved in this, really look forward to that time that the FDA releases that, because we want to meet FDA's expectations, and it would be great to know really what's expected from that perspective. With respect to exhaustive extractions, this is something that the FDA has been really rigid on. Even when in the past we were able to provide some explanation of why an exhaustive extraction wasn't scientifically making good sense, in specific cases, I see the FDA really expecting that now, for sure, for prolonged and permanent devices, and also in some cases with limited contact devices, as there's some residual concern with how long extractable chemicals might persist inside the body after exposure. So I think that going into this, you really have to anticipate doing an exhaustive extraction for your E&L studies, at least until we make some headway with the FDA on adding some nuance to that. Yeah, and you can see that this comes recommended in the new 10993-18. For prolonged and long-term devices, the recommended conditions are exhaustive, although credible alternatives include exaggerated conditions for those two categories. And I would say, from a scientific perspective, there certainly are cases where an exaggerated condition makes more sense. As you go to do an exhaustive extraction, it's kind of one of these situations where there isn't a whole lot of ways to do this that isn't going to receive some sort of scrutiny from the FDA. Non-volatile residuals is still the main way that people use to demonstrate exhaustiveness. This is the way that the FDA currently recommends. That said, if you do non-volatile residuals, you need to be prepared to address the question with the FDA of, why don't the masses that you measure when you do NVR match the total masses that you measure when you do GCMS and LCMS? And so this is a very common question. I gave an example there. It says you state that the total mass of NVR from the water extracts is 40.1 milligrams. However, based off of all the information provided, the total extracted amount was 3.12 for semi-volatiles, 7.42 from non-volatiles, and on and on and on. And then the FDA says that there's 81% of unaccounted mass there. So this is a common exercise. In truth, from the perspective of chemistry, these two amounts really shouldn't match. So if you do an NVR and that matches what you measure exactly by GCMS and LCMS, then I would be highly, highly suspicious that something, something's up. Those two numbers really shouldn't match. But still, we have to be ready to explain that if you do exhaustive extractions and submit this to the FDA. I think that better than NVR is to use things like TOC, THC, and ICP-MS to demonstrate exhaustiveness. We've been doing that recently in some cases with success. And then another option would be to do a full workup at every 24-hour interval until each chemical is observed to meet exhaustive conditions. And this is something that has also been well accepted by us and other large manufacturers that we work with. So I think that the third bullet there, the full workup, is probably the most ideal from a scientific perspective, but it also requires the very most work to accomplish that. The FDA very recently has been asking a lot about method suitability. This is something that historically E&L labs for medical devices haven't been upfront about sharing. A lot of labs have this information there, so they know, or they have some evidence in-house that their methods are suitable for the task that they're pursuing, but it hasn't been shared. And unfortunately, for a lot of labs, a lot of other labs where we done the toxicological work, we see that actually that information isn't there, and sometimes this chemistry data has been bad. So I think that it's fair for the FDA to be asking for this. We want to protect patients from having decisions being made based off of bad chemistry data. At the same time, if you're conducting these studies or planning to conduct these studies, I would expect the lab that you use to be able to provide method suitability data either in their report or make it clear in their report that this information is available to the FDA if the FDA should ask. And that information should include things like method validation for a range of compounds. So there should be like a set of surrogates that they've demonstrated accuracy and precision on, matrix, spike, and recovery through the entire analytical process, including liquid-liquid extractions, and so on and so forth. Something that needs to be clarified with the FDA and it be included in your chemistry report is this analytical evaluation threshold. The analytical evaluation threshold is the level from a scientific perspective where you need to go through the work and effort of making a chemical identification and quantification of that compound. It's something that's always applied regardless of whether you've used the term analytical evaluation threshold, because eventually we have to cut off the noise. But this is something that the FDA wants. They want to see how low did you identify and what's the justification for setting that level. And that level should be based off of a dose-based threshold, the threshold of toxicological concern, and then having some additional uncertainty factors applied due to the uncertainty inherent in chemistry measurements. So this is a screenshot of the new ISO 21726. It outlines the threshold of toxicological concern for medical devices. You can see for limited and prolonged devices, this threshold is 120 micrograms per day. That means that when you do a chemistry analysis for one of those types of devices, you need to be sure that you're not missing chemicals that might be above that level. So you want to have some sort of evidence that supports that there couldn't be a chemical above 120 micrograms per day that you're not reporting. And that's really what it's all about. There's a formula for describing this inside a report. I've seen the FDA ask for this formula specifically. You can see the FDA feedback there where they want to know what dose-based threshold did you use, and also what type of uncertainty factor was used in extending that dose-based threshold over to an AET. Okay, I think that we're going to run a little bit short on time here. You all have these slides available to go through, so I'm going to just go through the last few slides pretty quickly so that we have a chance to answer some of your questions. The FDA has had a lot of focus on the details of how compounds are identified. You should make sure that your lab can describe this and that they're using a range of different ionization and identification techniques. There should be more than one piece of evidence that supports an identification of a compound. Otherwise, you can expect that the FDA will come back at that. And the reason why the FDA is scrutinizing this is because there's been all too many examples like the one that I'm showing here on the screen where a poor or a very, very quick identification can easily lead to a misidentification. So if the compound was like what's shown and you use an automated system like Mass Hunter to just automatically make the call on the ID, it would indicate that it was the compound on the bottom left. However, we know because we've injected this compound into our own instruments that it is actually the one on the right. So these automated systems can misidentify, and so the FDA is aware of that, and they want additional scrutiny and expert review of those IDs in order to make sure that they are good. That's why having a database like what we have in Nielsen Labs, we have this huge database that we built slowly over time over the last 20 years, injecting compounds as they come up frequently so that we can positively identify them based off of the experience that we have with that compound on our own machine. And I think that this is something that the FDA is really moving towards expecting. So there's a little bit more detail on identifications here. I'm going to breeze over that a little bit. Suffice to say that the FDA really wants a lot of detail reported on how those identifications are.
Can I add something?
Yeah, please.
The one thing I just want to kind of mention on this slide is that designation as unknown. That's been a lot of discussion right now in the industry is what do we determine as an unknown compound and how do we deal with those unknowns? So please be aware that if you are submitting data to the FDA, especially if you're looking to justify out of one of those systemic toxicity endpoints, you have to be careful with compounds that are either tentatively identified, so you just have a partial structure or a partial match, or you have a complete unknown and they're above that AET. You have to be careful. You have to go through the work to be able to deal with those unknowns. So it's not good anymore just to submit a test report to the FDA and have a NIST and Wiley match with Mass Hunter like Matt talked about and says, this is our compound list. These are our tentatively identified compounds, and don't worry about these other compounds because they're just unknowns. We don't have anything to know about them. That doesn't fly. You have to be able to discuss and comment about any compound that's above the AET, regardless of how much information you have about it.
Yeah, that's completely right. It's really painful to submit chemistry reports right now with unknowns on there. We know that the FDA is going to dig in their heels on it, and I think we all have to be prepared prepared or have that be the expectation that if you have unknowns, that it's going to be a lot of scrutiny from the FDA. Yeah, with regards to reporting, like what they were saying here is that if you're going to use a class of compounds to make the identification, like if you say something like a C13 alkane, then what they really want you to do is list all the different possibilities. And if you stop to think about it, in some cases that makes sense and it can be done. But in a lot of cases, if you can identify something just as a class, really listing out all the members of that class is not a reasonable ask. And so this is where, there's been a lot of scrutiny that's been good, but it's also swinging a little bit too far and sometimes even impossible, as in the case of, the request to list all the members of a class. If you have something like a C32 alkane, for example, that's something that we would commonly identify as that, and you can handle that in a toxicological risk assessment. But to list out all 27 billion members of that class would be kind of ridiculous. Yeah. So really, right now the pendulum has swung, one way where the FDA is starting to ask for a lot. When we go to so the state of the art for us, if we go to execute one of these studies, you can see what our test matrix would look like. This sort of suite of extraction matrices and analytical methods is more or less standard for us at this point until we get further feedback from the FDA. Basically, we plan on doing the exhaustive extraction. Following exhaustive extraction determination, it's almost always exhaustive within 72 hours. Then we would do the real extraction on the medical device, 50 degrees C for 72 hours in 3 solvents, then execute using that suite of methods there. So this is what we would currently recommend. The FDA is expecting to see this method suitability data included in the proposal. So if you're planning on making a testing plan that you share with the FDA in a pre-sub, you should let them know that you plan on giving this type of information, and you should have this type of information in your reports. Things like calibration data and chromatographic data as well. The FDA is asking for a lot right now. The pendulum has swung full swing into skepticism. Sometimes to me, it feels like really they're asking for the moon, and I get why they might ask for that. They have a lot of skepticism. They want as much information as possible. I think it's fair to want the moon, but it's not realistic to give. And what we need to do is be able to provide them enough information that they can make a good call that's protective of patient safety. We can give them a small amount of that to get the job done. So just to step you through how we're doing this successfully, we'll report a lot of this stuff in our chemistry reports, and a lot of the same type of information is included in the protocols that we'll share with the FDA when we do a pre-sub. And then, so you can see, for example, it should be clearly outlined what the study purpose is. You have the absolute, like this very clear description of how you calculated the AET. So that's all delineated there. They want details on the sample preparation, so not just how you extract the device, but everything that happens to those liquid extracts between extraction and analysis on an instrument. All of that should be outlined in your protocol and report. You can see this continues here. There's more sample prep. Then, of course, the analytical procedures. There's a lot of detail there that we don't include in all of our reports, but we note that those procedures and protocols are available in full detail to the FDA or other regulatory body upon request. Then when it comes to reporting, we will give names of compounds, CAS numbers, retention times, of course, the amount in micrograms per device. Then you can also see the column there on the left. That's the level of identification. This has been an absolute requirement for the FDA. They want to know how good is the ID and how you know that it's that good of an ID. So so that part is required. There for us, an IC means an identified compound. That means that it's a compound that we've injected ourselves on our own instrument, and it's 100% confirmed. with respect to its identity. And then, of course, they want the chemical structures as well. All right, so just to wrap up and try to leave, I think 5 minutes for questions here at the end. So it needs to be understood that this chem-tox process is often viewed as a requirement for medical device studies. In the past, you really had, I think, more of an option to choose between animal studies and chemical studies. Today, we see some sponsors trying to not do any chemistry. They just do the animal studies, and you can get kickback from the FDA or other regulatory bodies asking for this type of chemical information. It's over the last 6 months been undergoing a really intense period of scrutiny. So we need to be prepared for that. It shouldn't be a surprise. The best way to address that is to have a really strong collaboration between the toxicologist and the chemistry lab. And I can't stress enough that you really should go through this pre-sub program right now if you don't want surprises at the end of your process. The FDA is changing very rapidly. So I highly recommend doing a pre-sub. We can help with that if that's something that you want. And of course, the goal for all of this is to save animal lives, to save people, improve patient care, save time and money. So it's a win for everybody to do this right. And with that, I think we have time to take a couple of questions here. I've seen a few pop up on the panel. Maybe Thor or the moderator can pick a couple of those, and we'll go ahead and address them.
Yeah, absolutely. So as far as some of the questions go, one of the questions has been, is the FDA seems to be more strict than ISO. Will following the FDA approach also fulfill the requirement for submitting in, for example, Europe?
Yeah, I'll answer that one, even though Matt can add in. So right now, the FDA, strict is an interesting term, but what we've seen is the FDA maybe has a higher skepticism with the chemistry that they're getting. So they have higher requirements for what they want to see. What we've seen is that any test that's set up to meet the FDA requirements has not had any issues with notified bodies. Where if you do a notified body type of testing, sometimes the FDA will have issues with that type of setup. So usually our approach is meeting the FDA's prerequisites for chemistry, and then we usually have no problems with notified body.
Okay, we've got some very specific ones in terms of extraction solvent and a few others. But do you have any recommendation in case the material tested is soluble with the non-polar solvent and we cannot perform the chemical analysis?
I don't know what you're talking about. Every single material reacts really well with hexane. I'm kidding. Matt, what do you want to?
Yeah, so this is a common issue, right? So the FDA and the 10993-18 is specific about at least attempting certain solvents. So, and oftentimes we know ahead of time before we even test that a solvent is going to degrade or dissolve a device. And so the approach there has been to, you have to still try those 3 solvents. What we'll do is an extraction feasibility study where we put the device in those solvents, and if hexane dissolves it, we take a picture of it dissolved, and that's demonstration that that solvent is not suitable for the study. And with that piece of evidence in hand, then we've seen the FDA be accepting of alternatives to that don't degrade or damage the device. But what you can't do is just skip to the alternative solvent. You really have to show or prove that it was affected. So if hexane dissolves it, then you could go to just 2, like water and IPA, or some alternative in between there.
Okay, we have time for probably just one more. Is it necessary to execute the E&L testing at different time points over the entire shelf life of the product, for example, at the beginning and end of the shelf life, or is one determination enough?
So I'll give some thoughts, and I'll let Dr. Jorgensen also give some thoughts. So it's my favorite answer to any biocompatibility question, and that is it depends. It kind of depends on what you want as far as information from the chemistry. If you're looking at just biocompatibility and you feel like your device is pretty stable through its shelf life, so if you're looking at a very well-known plastic or metal, and you have a pretty good sense and can justify that your material is not going to change, or if it does change, it'll probably just get better because you're probably losing volatile compounds over the shelf life, then just doing the E&L at the 0 times 0 would probably be the one that you would need to do. If you're wanting to prove that like a drug compatibility, or maybe your device or materials in your device could degrade over time with UV exposure or just time, then you would want to do multiple time points throughout the study, because you'd want to be able to prove, it's all about patient safety. So you want to be able to prove that your device does not get worse during its shelf life. So for the majority of devices out there that are dealing with polypropylenes and polyethylenes and metals, then the shelf life, we pretty much know what happens to that device in the shelf life. And so times 0 is pretty good. But if you are dealing with a material or a type of device that could have impact over time, changes during the life cycle, we'd want to put that in the test.
Yeah, these are great questions. So I agree completely with what Thor said. The only thing that I might add, just to be like specific about it, is that if the device is intended to degrade or change, then it is a requirement to measure it at multiple time points. That's usually the exception, because normally it's not intended to change once it's inside the body. But
okay. Thank you guys so much for the presentation today. We still have obviously quite a few more questions that have come in. Unfortunately, as usually is the case, we are running out of time for today. So anybody who submitted a question where Matt and Thor were unable to get to those, don't worry, they will be getting a list of all of these questions. They will be following up. Don't forget, there is a handout where you can get a copy of their slides. A recording was done for this, so you will be able to go back and attend and rewatch this session at a later point in time. There are other sessions going on. This is day 2 of a day 3 summit, so feel free to sign up and attend all of the other sessions. Thank you again, Matt and Thor, for going through all of this. It was a really great presentation.
our pleasure.
Thank you.
Thanks, everyone.